Wednesday, and welcome to this special longevity webinar. I'm your host, Dr. Amy Moore, Vice President of Global Engagement and Patient Partnerships. Today, we're very excited to have this special webinar where we're talking about a large new research initiative at the University of Michigan, funded by a generous gift from Judith Tam through the Richard Tam Foundation, looking at mechanisms of ALK resistance and efforts to develop new interventions for people living with ALK positive lung cancer. I'm joined by the team behind this exciting new project, including team lead, Dr. Sophia Maraver, Dr. Peggy Sue, and Dr. Angel Chen. We're also joined by Amanda Nerstad, who is the Vice President of ALK Positive Incorporated. So welcome to all of you. We're so pleased you're with us today. So I want to start with you, Dr. Maraver, to give us some history and background on how this project came to fruition. And by way of background, you're a professor of epidemiology in the Department of Internal Medicine at the University of Michigan, and also director of the program for breast and ovarian cancer risk there. So I'm curious to hear how you've leveraged your background in breast cancer to now go after lung cancer. So we're looking forward to hearing about your work and to the exciting research that you all are doing. Welcome.
Well, thank you very much, Dr. Moore, Ms. Nerstad, and all to everyone in the audience, patients, advocates, family members, researchers. It's a tremendous honor for us to be here and tell you about this program. Actually, my career in oncology started when I was 5 years old, and it came in contact with disease in general and cancer in particular. And then when I was 15, I lost my dad to lung cancer. And so lung cancer was forever my first love of a disease to cure. A series of circumstances took me to other cancers, and I've been incredibly honored and privileged to work in bladder cancer, melanoma, and now I am joined by my colleagues in a large group of people focusing on ALK-driven non-small cell lung cancer. If I could just share the slides, we have a small presentation to start with, and we look forward to answering all your questions. This is a bit of a dream come true for those of us involved in clinical research, translational research, and basic research, because through the generosity of Ms. Judith Tam, Sargent, we have come to be able to craft a project that really brings all the disciplines involved in state-of-the-art, cutting-edge cancer research in general to lung cancer. I think the efforts in lung cancer have been monumental, and a lot of progress has been made, but it is a monumental challenge also. And so this particular endeavor will really allow us to focus in great detail on the mechanisms of ALK-driven lung cancer from the beginning of the disease. How does the disease under treatment escape sensitivity to the drugs? How does progression really occur? And how can we invent new mechanisms and new modalities of treatment, both utilizing existing drugs in different ways, repurposing existing drugs that have never been used for ALK-driven non-small cell lung cancer, as well as inventing de novo new drugs. We are privileged to be able to do all those things under this initiative, thanks to Judy's generosity. And we are beginning to receive other funding, such as NIH funding, to study specific sub-projects derived from this. So today, I'm just here sort of opening up for the real stars of the show, who are my colleagues, Peggy Sue and Angel Chen. Angel is the lead clinician, and Peggy Sue is the lead basic scientist. So I'm gonna advance the slide. Hopefully I can advance it. I'm not sure. Okay. Okay, I think I advanced too many slides. Okay, there we go. Sorry about that. All right, so as I alluded, the Judy Tam Initiative has 3 major projects. The only way to execute these projects is by engaging with patients, by receiving a lot of feedback and collaborations from our advisors, both internal to the University of Michigan and external from institutions who have excelled already in work in ALK-driven non-small cell lung cancer, so multiple advisors and collaborators working as a single team. So this is definitely team science. We are not concerned with territories, with whose idea it was to do what. We are concerned about curing patients and coming up with new strategies to make patients have better lives, longer lives. So Project 1 is a precision drug evaluation project of patient material obtained directly from the operating room, from thoracentesis, and hopefully in the future also from circulating tumor cells, perhaps even small biopsies. As time goes on, we will perfect some of the technology to be able to expand the types of samples we can test a lot of drugs on to ascertain which is the best drug for the patient today at this moment. Project 2, led by Dr. Peggy Sue, who will speak shortly, is all about mechanisms, all about the basic science of how this disease starts, how it progresses, how it reacts to separate therapies, how does the immune system monitor the whole thing, and can we engage other elements, not just the cancer cell, in trying to advance our strategies in this disease. Project 3 is entirely new therapies, whether it be novel combinations of existing therapies, as I already said, repurposing new drugs, or inventing drugs. Project 3 is all about the drugs, better drugs, and overseeing all of this effort is Dr. Angel Chen, who is the clinician, the mastermind clinician for us, and trying to find all kinds of opportunities to engage with patients in all sorts of different research projects as the science advances. So that is a very big part of why we are here. So I wanted to tell you a little bit about Project 1 because it's of great interest to the community. We are honored to have received a lot of inquiries. So I want to manage expectations to some extent and at the same time beg for your help. So, you know, we live in that world. So what we want to do is to develop and validate a real-time drug testing platform for ALK-driven non-small cell lung cancer. In the process of doing that, we are very likely to also be perfecting a platform for non-small cell lung cancer in general, because we are also testing other lung cancers that are not ALK-driven, because ALK-driven disease is so rare. We are still developing a lot of technology, and other lung cancers are coming into play as well. Right now, we are in the process of obtaining all the regulatory approvals at the University of Michigan, but we are not yet seeking approvals outside. I am in the process of figuring out what are the best strategies when patients are located outside of the University of Michigan or outside of the state of Michigan in our immediate area. We have close collaborations, but not yet with the outside, but we are working on that. That's a challenge that we know we can overcome. We are collecting and testing samples coming from our own patient population. We would like to compare outcomes for patients treated in different ways, in different locations, patients from all walks of life, with all kinds of histories, trajectories of their health, ages, ethnicities. We'd like to standardize all these processes so that there would be a lot of confidence that when we have a panel of drugs that have been tested, that the patient and the clinician making that decision of what to treat next with will have a lot of confidence in our data, the way you do when you do your next generation sequencing, for example, and you find out that there is an ALK fusion type 5B, you and your doctor know what that means. That doesn't tell you what drug to use, though. And so we want to get to that next level where we use the actual cells from the patients to understand what drugs are most effective. And as time goes on, probably in a year or so, we think we're going to be in a position to begin to apply this technology under protocols where the patient and the clinician will be able to have this information in a systematized manner. We will never take that step until we are absolutely sure of the validity, the reliability, and the robustness of our technologies, because we understand the meaning of that at a deep level. So, but we have to start somewhere. So this is where we are starting out now. Why are we doing this project? As you know, all of you in the audience have heard of the fantastic drugs that are available for ALK-driven diseases, developed against ALK functioning itself, kinase inhibitors primarily, right? So yet we know that resistance evolves and evolves under pressure of therapy, but what are the exact mechanisms? Many are known. There are new mutations that arise, or maybe they were present to begin with. So is there inherited resistance already present and diagnosis? In different patients, the mechanisms of resistance may arise differently. And as a result, what drugs to use to counteract those mechanisms may not be the same. Any one mutation doesn't necessarily mean there is a particular drug that will work in everyone who has that mutation. We all wish that were the case, but this disease would be cured already universally if that were the case. So we already know it isn't. So we need to really admit about our limitations of having mutations alone drive therapy. This is something, of course, we learned painfully in other cancers like breast cancer, who always leads the way in blazing the trail of making a lot of assumptions and then having to put a bit of a realistic contour on those assumptions. And however, we feel very strongly that all the work done in other diseases can be applied to lung cancer and shorten the time it will take by years, if not decades, to understand lung cancer at the level of trying to find new therapies in the setting of resistance. So we are very, very focused on understanding that. And now I'm going to pass the baton to Dr. Peggy Sue, who will talk to you about Project 2.
Thanks, Sophia. It's really nice to join all of you. I'm Peggy Sue. I'm an oncologist and a basic scientist. And I trained in oncology at Dana-Farber and Mass General Cancer Center before moving to Michigan in 2020, where I started my lab, which studies really how normal lung cells become cancer cells. And I was drawn to studying lung cancer because there's really an excitement in all the ways that we can treat the disease right now that it's brought benefit to many patients, but there's still a lot of work that clearly remains. So we as a group are really interested in studying the entire lifespan of the disease. And my work is particularly interested in what happens at the beginning. So many patients with non-smoking, with ALK positive lung cancer, want to know why they developed cancer. And we as oncologists also want to know. And in particular, we want to know if there's a way to detect the cancer sooner and to treat it earlier when it's still at a curable stage. So lung cancer really begins with mutations in a cell of origin. So shown here on the slide is a diagram of the cell types of the lung, from the airways to the alveoli where gas exchange happens. And there's an array of specialized cell types in the lung which perform a a really beautiful array of functions, secreting substances that serve as a barrier or defense against infections, oxygen uptake. And a few of these cell types are special because they're also called stem cells, and that they can self-renew and also give rise to the other specialized cell types of the lung, either during normal wear and tear or under injury. And it's thought that these stem cells are the cells that, when ALK is mutated and activated, become the cell of origin that gives rise to cancer. So in my research, we isolate normal lung stem cells from patients, and shown on this slide is actually a picture of some of these patient-derived stem cell cultures grown in the lab. The different colors represent different stem cell types. And we then activate ALK to understand some of the earliest changes in these cells. Next slide, please. And so this really includes everything from how the cells might change identity, how they rewire their metabolism, how they become sensitive to different drugs. And our goal with this system is to tease apart differences, not only with ALK and normal, but among the different ALK variants. We cross-reference studies in humans and mice, in vivo, in the patient, in the organism, and also in the cell culture dish. And we can also culture tumors from patients in a similar manner to study how it compares to their own normal cells. And so again, we want to study how cancer starts in order to detect it earlier, treat it earlier, and understand how to prevent it from progressing to a more advanced stage. And we can also take these cultures and introduce different cell types like immune cells, which we now know are important in fighting cancer, but perhaps are dampened in ALK positive lung cancer, and understand why that happens. And with that, I'll pass it along to Dr. Chen.
So my name is Angel Chen. I'm a thoracic medical oncologist here at University of Michigan. I did my fellowship here at the University of Michigan, hematology oncology, and I just love the mentorship that I have here, the wonderful team building that we have here. So I was lucky to stay on here. So I, as Dr. Mariver mentioned, I am the lead clinician on this research initiative, and my goal is to bring all this wonderful science that Dr. Mariver, Dr. Sue, and others are doing, and try to bring it into the clinic so that when I meet a patient sitting in front of me, I want to be able to say more concretely, what does this mean for you? What have we learned about your lung cancer? What are some of the things that we are trying to study, and hopefully through what we've learned, also develop new treatment therapy options, whether or not are we looking at combining different drugs from the get-go? Are we going to combine these drugs in later when we detect a specific mechanism of action, or a mechanism of resistance? And also, are there any other drugs either already existing but maybe not being used for treatment of lung cancer, or are there new compounds that are just kind of waiting to be discovered based on all this work that Dr. Mariver and Dr. Sue and others are doing? And that's really where I see myself as kind of like the face that the patients get to see and ask these really important questions about what we're trying to do here in our research initiative.
Wonderful. Well, thank you so much. Did that complete your presentation?
Yes.
Okay. Well, I mean, clearly this is a very exciting initiative that you all have started, and we're so thrilled for what it means, not only, I think, for patients living with ALK positive lung cancer, but I think a lot of the mechanisms that you're exploring have implications and can inform, other biomarker-driven lung cancer. And so I think, that's also something we need to appreciate and understand. So I know there are a few questions in the chat about your presentation, but before we get to those, I want to just briefly pivot to our final guest, who is Amanda Nerstad, who is a person living with ALK positive lung cancer and also serves as vice president of ALK Positive Incorporated. And I thought it would be critically important just to have the patient perspective on this initiative. I know there's a lot of excitement and buzz among the broader ALK community around the world about this. The word has gotten out. And so, Amanda, maybe you can offer some comments and perspective on what you're hearing and what this project means to you as someone living with ALK positive lung cancer.
Absolutely. Thank you very much for having me. My name is Amanda Nerstad, and I was diagnosed with stage 4 ALK positive lung cancer in 2016. And like all ALK patients, it was a complete shock. And thanks to research and, you know, every initiatives like this, we're so appreciative. I also want to say that, you know, we're very appreciative of Judith Tan's donation and the University of Michigan. This initiative team is great. I always want to have like plan A, plan B, and plan C in place for if and when I progress. And this initiative that you all are offering, it definitely on a physical standpoint, it provides more treatments or provides another plan for all of us. It extends our lives, hopefully, to all ALK patients and can continue making memories with our families. And mentally, it also just provides so much hope for all of us. And so we're so grateful to the University of Michigan team and also, you know, being diagnosed with stage 4 lung cancer 5.5 years ago, thanks to research, I am still able to make memories with my families, and we hope that this can continue moving forward.
Thank you, Amanda. And we also want to just give a nice shout out to Gina Hollenbeck, who has served as president of ALK Positive. And she, I believe, is the first patient maybe outside of the University of Michigan to contribute tissue for this project. And I know she sends her congratulations on the effort and appreciation for the work you're doing. I think it means the world to her and her family and brings a lot of hope to the broader community. So she sends her regards, and hi, Gina, we're glad you're home, and hopefully each day is a little bit better than the last. So we do have some questions that I will start. And one was about the first project, Dr. Moraver, and just thinking about how this work could potentially inform treatment sequencing. You know, that's something where there's a broader opportunity, I think, given that we have so many approved drugs in the ALK space. You know, something that comes up frequently is this question of real world data and patient reported outcomes. And, you know, there's a lot of interest in thinking about treatment sequencing and how we can, you know, approach that to achieve optimal outcomes for patients. So how does your work inform that conversation?
Yeah, exactly. Thank you, Dr. Moore, and thank you for the question. I saw it in the chat, and several people are asking that. So that is at the crux of the matter. Thank you, Amanda, by the way, for sharing your story, and we are very, very grateful to all the patients. So this is a project about each patient. This is a project of true personalized medicine. We want to be able to test the cells that are affecting your quality of life today. When they come out of the body, we want to be able to test them for a number of drugs. In order to do that, we need a fairly good sample size right now. You know, about 1 centimeter. Most biopsies done by needles are not going to be 1 centimeter. We can test fewer drugs with a very small sample, but to do the best work for the future, we will need a little bit bigger sample. So I'll talk a little bit more about some of the ways in which we may get around this. But right now, that's exactly our goal. We have, thanks to Dr. Chen's guidance and talking to a lot of collaborators at multiple institutions, some of which have already been mentioned, we are stopping at nothing to speak to people, to collaborate with people. We have every single drug, essentially. I mean, I don't want to be saying every single one, but I think we have every single drug used in the United States or Europe, or either approved or in clinical trials. And of course, we have many experimental drugs that are making their way into the clinic through phase 1 trials, or maybe just left the preclinical animal use stage into humans. Plus, we have our own compounds, and we are very open to testing compounds that other institutions may be developing because of our platform being so robust in terms of being able to test drugs. So, for example, a patient we would get, say, for example, a pleural effusion sample. Pleural effusions are obviously something that patients know very well affects their quality of life. So they often have to be tapped. They also often have to be evacuated. They provide tremendous numbers of cells on which we can do a lot of investigation about drugs. And so we would know the history of the patient. And so one of the ways is we would test them for all the drugs, but we would, without knowing, we know the history, but it's on a separate file. We would predict then the resistance to the drugs that they've already had, and at the same time, we would test other drugs, unexpected drugs perhaps, that might be in use in other diseases or in other types of lung cancer, and perhaps new combinations of existing ALK drugs that haven't been used before, or an ALK inhibitor plus something else, like a MET inhibitor or a MEK inhibitor or a MAP kinase inhibitor. So we are very open-minded because we know from our work in bladder cancer and breast cancer that there are many different pathways that cancer cells use. It's not possible to predict how they adapted to the presence of the first inhibitor. So so that's what we're trying to untangle. So even after we test the drugs, if some of the cells survive, we can test those cells against more drugs, so that the hope is to basically eliminate all of the clones. And most patients who have had disease for many years will have multiple clones. I think I'm not breaking any news to anyone on this call, on this conference, about that. And I know a 1 centimeter tumor can have multiple clones. So that is our perspective. We expect to be able to be challenged by patient material in terms of size, but we are trying to get around that in multiple ways. And one of the ways I already mentioned, access to pleural effusion fluid. The other thing that we are very focused on, and wish us luck, I think we are at the point that we are going to be successful, but I'm fingers crossed, is to expand circulating tumor cells. So the circulating tumor cells we can capture, no problem. Patients with ALK-driven disease have a lot of circulating tumor cells. I am surprised by the numbers that we are seeing, and so we want to be able to expand them, to be able to test them. Right now we are at that juncture of trying to expand them. So wish us luck. We think we are going to be successful.
Well, thank you. And we certainly hope you are. So a couple of questions. You talked about size of the sample, but I also want to think about size of the project. How many patients are you hoping to enroll? Do you have a set number, or are you just trying to enroll as many as possible?
No, as many as possible. So a lot of people are reaching out to us, and we are incredibly honored and humbled by it. We are answering everyone. We are developing a very small questionnaire, organized amount of information that we would like to get from everyone up front, such as obviously your diagnosis, your date of diagnosis, and your NGS, your next generation sequencing, including the actual report, your narrative, whatever you want to tell us about how you feel, how you're experiencing this disease. That's very important for us to know. It's not just inspiration, but it is part of part of what we're trying to do here, which is quality, right? And so we want to know how you are experiencing the different drugs. And so, but in the initial questionnaire, we are going to be asking for just a small amount of information, which is what I have been doing when I have replied to the emails. And then after that, we're hoping to then contact you and enroll people, whether they are having an intervention or not. I think we like to enroll and know when you are coming up for a CAT scan or a PET scan, and then are you going to have an operation? Are you going to have blood drawn? And we could try to opportunistically collect tissue at those times. I don't have a set number of patients. We are nowhere near enough numbers to be able to have enough experience with testing the drugs and understanding resistance. So no, if we don't work together, we are multiple institutions. On the other hand, this audience is very sophisticated in clinical research, and there are a lot of barriers to just, you can't just send tissue places, right?
Right.
There are a lot of regulatory, pathways that we need to make sure we transition through.
So there are several questions in the chat that do get to more of the kind of mechanics and logistics of the study and people who are interested in participating. So I want to try to just walk through those so they have greater understanding of how they might be able to participate. One, I know you touched on pleural effusion. So it sounds like you still require tissue. So if patients had only a liquid biopsy or blood sample, would they still need tissue to participate?
No, they don't. Not up front, but if at some point the tissue is needed. Plus, we could also, we have an interest in having some of the original tissue at diagnosis sent to us, and so that we can then understand what kind of clones, if we can sequence more deeply, or we can understand the immune environment. There's a lot of information coming out of Dr. Su's lab, other labs, Dr. Lagisetti, who's a thoracic surgeon. Other people are doing work on the immune environment. I have a new scientist starting in my lab in July who's going to completely focus on the immune environment. So we like to also connect to your original tissue diagnosis. And there is a question here about brain metastasis. Mm-hmm. Obviously, it's a very serious event in any kind of cancer, and we have a very powerful platform to study brain metastasis. So anybody going to the operating room for brain metastasis, that would be a tremendous opportunity, because our platform works incredibly well for brain metastasis of all types of cancers. That's been in place for several years, and we are writing a very big paper on that right now. And Dr. Morikawa, one of my mentees, is the lead clinician on brain metastasis. So we are very interested. Obviously, it's a condition we take very seriously, that the right drugs have to be given as soon as possible. In that situation, in all situations, but brain metastases have an extra level of urgency, of course.
So we do have some international viewers and international ALK community. So there's a question about whether or not you're able to accept a patient from Europe at this time.
It would depend on the regulatory environment in the country. I have done a lot of international work from Africa and the Middle East and North Africa and South America, and as long as we respect the regulatory environment in every situation. So it would have to be worked out, where our protocol would have to be either emergently or urgently approved, so that the tissue can be received. One thing that I want to warn people is, if you call me today that you are having a biopsy, in 3 days, we are not going to be able to receive the tissue. It's just not possible because of regulatory issues. But I am working with our IRB at Michigan, and there's a lot of discussions going on about our work because we are blazing a lot of trails, you know, I'm incredibly grateful for your patience and your understanding that we'd love to receive everything, but we want to make sure it's all done systematically. So when it comes time to say, this is the knowledge we have gained, that we can use your sample, because if it wasn't obtained correctly, we can never use it. And that would be a real shame and a betrayal of our trust in clinical research. So everything has to be done perfectly so that the knowledge can be gained for posterity.
So to that end, there is one question about whether patients would be able to have the biopsy at your center instead of their own clinic while covered under their existing insurance. Is it preferable for them to have the biopsy there so you can kind of ensure the integrity of the sample for this project, or can they have it at their own home's location?
Well, I'm going to let Dr. Chen answer that question because I don't like to be the center of all the questions.
You know, I think that that's a really good question. You know, I mean, we understand how difficult it is to travel to, you know, to Ann Arbor. It's beautiful though. You guys are really, you like it. But you know, it's not easy to travel, especially with COVID and everything, and of course time and money. So I don't want to say we definitely want to enroll as many patients as we can. You do not have to come see us in person, face to face, or get your procedures here just to be enrolled in the study. If it's feasible, then absolutely, I think that would be the easiest way to enroll, because then we wouldn't have to worry about the regulatory issues, because you would have been consented here at U of M. You are a U of M patient once you walk into the door, and then we can take the biopsy that would be clinically indicated. But like Dr. Mariver has mentioned, we are trying really hard to be able to receive the biopsies no matter where you are.
So I see there's I'm sorry, were you about to speak? I didn't mean to cut somebody off.
Oh, no, all I was going to say is I echo what Dr. Chen said, that we like to do what's best for the patient. And, you know, so we want to work it out. If it is convenient for the patient to come to us, it's a lot easier for the project. But we're going to work with the patient no matter where they are. We just need a little time to get more organized.
Yeah. So I have one question just about what stage you allow, and I see that you welcome all stages, so that's good. And I just want to, before I shoot a question over to Dr. Su, you know, we're seeing, at least in the EGFR space and in the immunotherapy space, you know, shifting of treatments to earlier stages. So we have, you know, osimertinib for earlier stage EGFR patients. We now have immunotherapy, both in the neoadjuvant and adjuvant setting for early stage patients. And you know, I come back to something that another ALK researcher once said, Dr. Trevor Bavona, who's at UCSF, and we had, when I was with GO2 Foundation for Lung Cancer, we had funded a joint collaboration between him and Dr. Christine Lovely at Vanderbilt, where they're looking at combination approaches to try to ward off resistance. And, you know, something he said really stuck with me, and it's, you know, why do you want to kind of interrogate the black box after the plane crashes? You want to keep the plane from crashing in the first place, you know, so trying to hit tumors hard and early to prevent that emergence of resistance. And so, you know, that's where I'm thinking about the implications of this work in the early stage disease setting, and how you can begin to understand those very basic biological mechanisms that may point to emergence of resistance clones. So perhaps you can speak to some of the work you're doing, Dr. Su, with these stem cells and what it may suggest in kind of the earliest stages of resistance mechanisms.
Thanks for that question, Amy. I totally agree. I think what we're learning from other cancers, particularly like EGFR, is I fully predict that in the future, we'll be moving our ALK inhibitors earlier and earlier. And I totally agree. I think there's a really beautiful body of science now that suggests that we should be almost preventing resistance rather than treating it after the fact, and potentially treating upfront with more potent combinations rather than waiting for a specific mutation to come up. And that's definitely something that we're planning on modeling in my stem cell cultures. I also think, however, that there will be differences between, for example, EGFR positive lung cancer and ALK positive lung cancer. We know that, just the clinical courses are already different. And I think there are things that are happening very early on that might also be different. Specific hypotheses about how the cells change upon ALK mutation. And so I think we're learning a lot from the other oncogene-driven lung cancers, but I also think that ALK-positive lung cancer will potentially be a unique case. There's also a really fascinating paper which suggests that potentially these fusion-driven lung cancers may have arisen, in many times, many years before patients develop lung cancer. And that may also be different between, for example, ALK or RET versus EGFR, that there could be a long latency. And that could be in the future a potential time where we can really intervene, given that, that ALK mutation could just be sitting there for a little while. And so I'm really optimistic that we'll be able to study that early state and hopefully prevent it from becoming more, from progressing. It's a really great question.
You touched on, and I know this probably resonates with Amanda and some of the other viewers, being, relatively young, to have a never smoking history. Do you have any hypotheses yet on, maybe some of the mechanisms behind emergence of ALK lung cancer, or, will the mechanisms look different in that population versus maybe an older population, or is it too early to speculate?
I think it's a bit early. It's a wonderful area of interest. I think it is a bit early to really speculate. However, I do think that there are differences between, stem cell function of the younger person versus the older person. And there is a reason why, cancer is kind of typically a disease of the elderly. However, with these non-smoking lung cancers, we're really seeing patients who are getting it very young. And so I think there is an interesting area of biology to explore there, the young stem cell, the old stem cell, and then what happens when you have ALK mutation or other mutations like KRAS. But it's still very early, I would say.
Mm-hmm. I think there's a lot of interest. You spoke to really understanding the interplay with the immune system. And I know ALK Positive and Longevity have supported research projects really trying to unlock the potential of the immune system, by way of vaccines or other approaches, as an addition to the armamentarium of options there. And so do you anticipate, combinations that would, kind of pull from the TKI resources along with immune interventions? And do you have any hypotheses that you're pursuing yet on that front?
I think that's a really great question. I think it's also obviously an area a lot of patients are interested in, right? Because you see, on TV all the time, advertisements for these immunotherapies. I think, though, what we know more and more from the EGFR and ALK world actually is that single checkpoint inhibition with drugs like pembrolizumab, nivolumab really doesn't seem to work or have an effect on ALK positive lung cancers. And the question is why? And some of it can be because ALK positive lung cancers we're finding a lot more frequently in young people who don't have a smoking history. So there's that traditional thought of, a cancer induced by or characterized by a lot of mutations from a strong smoking history. So your immune system doesn't really have a great target, so to speak, to go forth and seek out. But it doesn't mean it's not possible, right? I think your immune system, as we have clearly seen, is this incredibly powerful tool that you just have to learn how to harness appropriately. And what does that mean for ALK positive lung cancer, I think is definitely an area of interest, because if we can somehow harness the power of the immune system to go after ALK positive lung cancer, that would be a huge advance in this field. And actually one of our surgery colleagues and collaborators, Dr. Lagasetti, is actually interested in this very specific idea of what the immune microenvironment might look like in someone who has an ALK positive lung cancer. What is the interactions of these different immune cells with the tumor itself, and is there something there, that we can either harness or disrupt the engagement of to really make the immune system active? So I think that's an area of very hot research right now.
Amanda, I know you've been putting some questions in the chat, but are there other things that you're hearing from the broader ALK community with regards to either this project or concerns that you think this project is particularly well suited to address?
I think everyone is very, very excited. And the only concerns are how do I get involved? How do I become a part of that? And also, is there a limit? Are they taking a limit? So it sounds like you've answered those, which is great. But just making sure there's not a limit of number of patients.
No, not right now. We are nowhere near the limit, and we'll keep learning and enrolling people until we run out of money. And well, I mean, that's the reality, right?
Right.
So this work isn't inexpensive, and the way we're doing it, which we are attacking all 3 projects at once, and we are basically just not stopping on anything. We are getting all the models. We are synthesizing all the variants that we come in contact with, and the ones that we don't come in contact with that are already in the literature. So, we really want to get solutions for patients, and we want to accelerate the time it would have taken, apply to the NIH, then wait 2 years, then get a so-so score. So we don't want to go that route anymore immediately for this project. We're very grateful to the NIH, in case there is any people here that are sitting on study sections. Thank you very much. But we already got an NIH grant that we are expected to be paid, that we will study ALK-driven brain metastasis as one of the major initiatives of that grant. But we just need to make progress faster, I think, and that's what we promised Judy we would do. So that's what we're doing. We are working very, very hard, and we are limited by the number of hands we have, and through our collaborations, we're hoping to increase that. And our collaborations with the patients, we will learn nothing without the patients. So thank you all for your number one is the inspiration. I have never worked in a field before. I thought I had seen everything in medicine, to be honest, after so many years. But I am completely in awe of what I see in the ALK community and in the lung cancer community, which I'm embarrassed to not have known about, but now I do. And it's just completely, it's redefining how we are thinking as a group about what to do. It's the patient's enthusiasm, the patient's interest. It's just truly redefining how we are approaching this work. So we're not saying this lightly. Part of rethinking, I never thought I would find myself trying to enroll people in 50 states of the United States and in Israel, in Europe and Africa. But if that's what it will take, we'll do it. I promise.
Yeah, I think what you said is really a testament to the lung cancer community, because as we've understood more and more about the molecular underpinnings of the disease, we've seen groups like ALK Positive kind of rise up, and they're very committed to partnering and investing in research. And there are many groups like them who are doing the same, and we're seeing the the outcome of that, in terms of improving outcomes and survivorship. And we're beginning to chip away at lung cancer mortality and see increased survivorship because of advances in research and TKIs and immunotherapy, and there's much more to do. I did have that question, and I think you just kind of answered it for me, Dr. Moravir, about how you're addressing all the different variations, but it sounds like you're engineering those or creating them in the lab. So to address all the variability in terms of the different fusions and fusion partners. So that I think is critically important, just to make sure you capture all those different avenues. But I think the other thing that really resonates with me is just the team approach here. And you really are, tackling this problem from the very kind of basic research point of view, which resonates with me because I'm trained as a basic scientist and used to work on Wnt signaling back in the day, and with cell lines a lot in animal models, all the way through to the clinical translation of the work and engaging with partners, near and far. So I think, I'm excited about the potential of this project. And I know that with the support of the ALK community and the larger lung cancer research community, I think, there's so much potential there. But I know we're coming up on our final minute. So I just want to give the opportunity for closing thoughts. And Amanda, maybe I'll start with you and then ask your team again, what you need next and what would be most beneficial. But Amanda, would you like to offer some closing thoughts or final questions?
Yes, well, absolutely. I would just want to reiterate also how appreciative we are to all the work that you all are doing. And we're really excited. The ALK community, we all cheer each other on. And, I think a theme that we always talk about is we're all stronger together, and not just ALK patients together, but, having partnerships and working together with organizations and other cancer centers. So we're so appreciative. And just my own little plug is like, please remember, all the slides that you're looking at, and the cells and everything, that those are all ALK patients with families and kids, and you're helping them, continue memories, and we're all so grateful. And I just can't extend my appreciation, and everyone is so grateful for this initiative. So thank you all so much. And we really have a lot of hope for this project, and to continue more research and more life, for sure.
Thank you. Thank you.
And now finally to the three of you, final thoughts, comments, points you'd like to make.
Well, I like to start with saying, how privileged I am to have Judy Tam as my friend, and to have the opportunity of make a promise to a dear friend of mine, at the same time as realizing an old childhood dream of working on the disease or the set of diseases that robbed me of my dad when I was only 15. So this is just an incredible privilege, and we don't forget for a moment that we are looking at somebody's cells. That is somebody who has a life to live, and that's what we are devoted to. Thank you very much for this opportunity. I want to pass it on to Angel, who sees the patients day in, day out.
So again, I love that comment, Amanda. I think it's really easy to forget when you're looking at cells or looking at a specimen or some blood, that you're like, that you forget that there's a person associated, a very real person with, their own families, their own life's journey of how they got here. And I think that to me is so important to recognize too when we think about therapies, because yes, absolutely our goal and hope is to get rid of this cancer, but we also 100% need to make sure that cost is not too great, right? We want to make sure that there is a quality of life that is preserved. We want to make sure that you can continue to build these memories and build more memories, and that you're not, sick with, whatever concoction that has come up, right? So I think that is really important. That's what I really stress with my patients every single time I see them in clinic is, well, let's talk about your scans. That's great. But let's talk about how you're really doing, right? Because your blood counts and your liver function, kidney function, they look great, but how do you actually feel? And I think that human piece is is actually what I love. What about what I do, which is seeing patients in the clinic. And so our hope is to have, more great stories and longer stories for all of us to share.
And Dr. Sue, what does this project mean to you?
Wow, I couldn't have said it better than Angel just said it. I think we're really inspired by all of you as patients to work really hard. And I will promise that we will do that, because our goal is really to cure ALK positive lung cancer, either by detecting it earlier, preventing it from progressing, to treat resistance. And, we're really motivated to do better for all of you. So thank you for inspiring us.
Well, and it's not lost on me looking at this screen that there's a screen full of powerful women who are really dedicated to making a difference in the fight against lung cancer. So, as a woman scientist, it really excites me to see such a formidable team here. So we appreciate all that you're doing, and we're excited to see where this goes. And we're looking forward to lending our support and the support of the broader ALK community. So before we wrap up, I just want to remind people that if you haven't checked out Longevity's new lung cancer patient gateways, we just recently launched our ALK specific patient gateway, and that's where you can find resources about ALK positive lung cancer, as well as a map of specialists. So you can find experts who treat ALK positive lung cancer. There's information about clinical trials. You can connect with others like you through Longevity, as well as other ALK groups like ALK positive. So we encourage you to check that out, and we're looking forward to your progress. So thank you again for making time. We know how incredibly busy all of you are. Thank you, Amanda and ALK Positive, for your support of this event and of the broader ALK community. So with that, I'll conclude our webinar, and we look forward to seeing you again in the future.